Hello, my good friends out there. I remain Dr. D.I. June, your lecturer on chemistry on social media. But as I said before, I'll be treating some few topics on biology, and thereafter I will switch over to chemistry. So please, when I finish, do like my videos, share them to your friends, and subscribe to my YouTube for more educative lectures. So let's go. Our previous lecture on biology was the reasons why Mendel chose the garden pea plant for experiments. And I give you some of the parameters that made him to do so. But right today, we talk about the methods that Gregor Mendel used in his experiments. So now listen carefully to what I have in my note, then we'll explain step by step. Gregor Mendel used two major methods in conducting his experiments. These two methods include monohybrid inheritance and two, dihybrid inheritance. So this time we are going to discuss the first one titled monohybrid inheritance. Monohybrid inheritance. Now listen carefully. Mendel used artificial method to crossbreed two different plants at a time, which differed in one pair of contrasting characters. For example, tall and short plants. Tall and short plants. These are contrasting characters. Okay. Now, this procedure was called a monohybrid inheritance, and it was an example of a complete dominance case. We have dominance and recessive. If you were listening to me when I was discussing about the terminologies in genetics, that's why I said those things you must know them first before we advance. Now, he carried out the experiment in the following stages. Stage one, what did he do? He planted all tall plants, pea plants, for several years and discovered that the plants all produced were all tall plants. In the same way, he planted short pea plants for also several years, and he discovered that all the plants that produced were short pea plants. Now, in stage two, he proceeded to plant both the tall plants and the short plants. By the time flowers were produced, he now collected the pollen grains of the tall plants, tagged it as the male, and he pollinated the stigma of the short plant, which he tagged the female. Then he also collected the pollen grains of the short pea plants and pollinated the stigma of the tall pea plants. Now, Mendel then covered the artificially pollinated flowers with small paper bags to prevent the chance of natural pollination by insects or birds. After he did the cross-pollination, he covered the flowers with polythene bags, because normally we have what is called pollinating agents, either insects or birds, even the wind. Now, to prevent pollination by other insects or birds or wind, he covered them up. Now, in stage three, what did he do? Mendel once again picked the seeds formed after the cross When he planted the seeds, the plants obtained were all tall plants. These is referred to as the first filial generation, which we abbreviate as what? F1. Now look at it here on the board. This is figure A. Now, the first experiment that all the plants were tall. In this stage 2, all were short plants. Now, what he did was that, now look at the first filial generation. All were tall. Now, this is the phenotype, tallness for the male, shortness for the female. The genotype, the genes they carry, this is capital T, that is tallness, pure tallness. Small letter t, small letter is pure shortness. Now, when this gamete meets this gamete, it will have capital T, small t. When this same gamete attacks this one, it will have capital T, small t. Then this also will attack this, will give you capital T, small t. This again will attack this, will give you capital T, small t. You can see that the capital T is dominating. So, though they are heterozygous, but having what? A contrasting gene. So, the phenotype, that is the physical appearance of them, are all tall, but they are all carriers of this short gene. Now, what he did in the second is that he now took one of the tallness and another one of this female side. So he now crossbreeded two of this type. You can now see the male and the female. Now, after crossbreeding, you will now see that you have T, double T, then T, small t, small t, then pure shortness. This is what we call the first filial generation. The first filial generation. Let me put it in a local way. When a woman and a man marries, the direct children they produce are referred to as the first filial generation. All right. So we'll take that one as figure A. Then in stage 4, what did he do? Mendel then crossed the first filial generation plants and collected their seeds, and then he sowed them. He planted them. Then what he got from the planting was that these were tall and short. Tall ones were there, short ones were there, to the ratio of 3:1 respectively. So he called this stage of plants the second filial generation. That's what you are seeing here. He took the seed of the tallness, took the seeds of shortness, and he cross-planted, or cross-breeded them, and discovered that one was purely tall, like this one here. These two are like the first filial generation, while this one is purely short, as the female in the first case. So he now called this set of products the second filial generation, which we abbreviate as what? F2. Now, if you look at the ratio of it, 3:1. Look at it now. This is tall, this is tall, this is tall, 3. Then is 1. In other words, this one is purely tall. There's no carrier of short gene. These ones, they are all tall, but they are carriers of the short gene, small t. But this one is typically pure shortness. All right. Now let us see his observation. The outcome of this experiment leads to Mendel's first law of inheritance. This result that you obtained led him to his first law of what? Inheritance. Now, note that in figure A, the genotype of all the F1 is capital T, small t. Look at it now. In the first diagram here, which is figure A, the phenotype here, all is tall because capital T, capital T, capital T then, but they are all carriers of the short gene. Again, the phenotype of all the first filial generation is tallness, but they are what? Heterozygous. Look at it here. Heterozygous. Capital T, small t, capital T, small t. That means they are not homozygous, but hetero. I mean, the tallness is there, shortness also is there. But in the phenotype, the physical appearance is that they are all tall, but the gene that carries shortness is in them. That's what it means. All right. Now, to obtain the second filial generation, which is this one now, what did he do? The F1 plants, which is the first filial generation, this one's here, he cross-pollinated them. This is what I'm seeing there. And this is the result, which is now the second filial generation as illustrated in the figure B in this way. And this resulted to one pure tall one, two tall one but carriers of the short gene, and one pure short one. So this is how he made this result. Now from here, he made his first law of inheritance. But right now, share this video, like it, and please subscribe to my YouTube as we go to the next lecture when we meet again. Thank you.